Welcome to Ask the Expert. This is a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event. We're going to post it on the Sugar Science site YouTube channel shortly after the presentation. If you have any questions for our guests, feel free to enter them in the chat at any time or raise your hand at the end of the presentation. So today we have two guests coming to us from Copenhagen. And our first guest is Dr. Christina Pedersen. She's a postdoctoral researcher at Bartholin Institute, Rigshospitalet in Copenhagen. And she has been working with the 2 to 5A system on both DNA and RNA levels for her entire career since her master's, first in cancer and now in type 1 diabetes. We also have Dr. Karsten Buschard. He's a researcher at the Bartholin Institute in Copenhagen as well. He invented or described various prevention areas, which include low-carb diets in the 1980s and gluten-free diets in the 1990s. In 2018, he was an author on the use of fenofibrate, on a paper that described the use of fenofibrate to induce remission in a newly diagnosed type 1 diabetes patient. This patient has been in remission for 3 years. And so we'll kind of touch on that. His focus is really on sphingolipids in type 1 diabetes, and this focus has been really ongoing since 1990s. The paper, or the title of the talk today, is Dysregulation of the Innate Antiviral Immune System in Type 1 Diabetes, Prevention and Early Cure. And welcome to you both, to Christina and Karsten. Thank you for joining us. Can you give us a thumbnail sketch of your career path? What excites you most about the research you're working on right now? And then we're going to sort of launch into the paper. So I'll have Christina go first.
It's definitely to see or to be able to kind of contribute with of course new knowledge and basically kind of presenting how important the innate antiviral immune system actually is in the pathogenesis of type 1 diabetes. And I can't wait to dive even more into it. I'm relatively new in this area, but it's gonna be fun, which is not a good thing to say about a disease, but it's going to be interesting research that's going to come out of it.
Fantastic. Karsten, would you like to have a few words about sort of your background? What are you excited about right now?
Yeah, I'm most excited about the use of fenofibrate, which we hope could even improve the remission, maybe if started early enough to prevent diabetes. And I have been working in prevention and early cure of type 1 diabetes my whole career. We have a lot of type 1 diabetes in my wife's family, so I know about it. It's really a goal which I hope I can achieve before I'm going to retire, which is not now.
Fantastic. Can you just make a quick comment on what is it about fenofibrate that just a sort of a big statement about, what is it about fenofibrate that is interacting with the type 1 diabetes etiology?
Fenofibrate increases the amount of sulfatide, which seems to be a very crucial chaperone for insulin. It folds insulin, it preserves the insulin crystals at low pH, and when insulin is secreted, sulfatide is facilitating exocytosis and monomerization. One molecule of insulin is also facilitated by sulfatide, and fenofibrate increases this compound. Also, fenofibrate is an activator of PPAR alpha and has several other properties. But these sphingolipids seems to be very, very important for the beta cells and maybe also for the entire disease. So that's my main interest at the moment.
Okay, great. And would you say that some type 1 diabetic patients that begin, experiencing symptoms, are they experiencing a deficit of sulfatide?
Yeah, it's we can't measure it regularly in the pancreas, of course, but we have access to some samples from NPOT and from the Norwegian study DIVID, and there we have found and published that there's a lower amount of sulfatide in these pancreases and islets, and that fits well with the idea we had about how it starts and so on. So yeah.
So is this decrease in sulfatide systemic or just specific to the pancreas?
It's most likely also systemic, but at least we have found, or most importantly, we have found it in pancreas, in the islets of Langerhans, and we have found polymorphisms in some of the enzymes that create these sphingolipids, and we have also found these enzymes low at the mRNA level in these samples from newly diagnosed type 1 diabetic patients.
One more question. If you had if it's a systemic issue with a decrease in sulfatide, wouldn't this impact cells in the gut, in the small intestine?
Yeah, could well be. But it seems that these sphingolipids are really important in the neural system, the brain and the peripheral nerves. It's a compound, sphingomyelin, which is isolating the nerves, and it seems to be very important in the beta cells, which are derived not from neural cells, but are influenced from neural cells, so to speak.
Yeah, they have some similarities in their lineage.
Yes.
Well, let's dive into the paper, and let's have Kristina, could you present? Are you able to present a couple slides here? I think yep, it's all ready to go.
Yeah. Is it possible to do it with just portion of the screen?
Sure. I think if you go down to share screen and then you get your slides up, it should go right to, yeah, perfect.
I just need to, 1 second.
The size might be a little bit, yeah, that looks great.
Perfect. Yeah, so should I just start or?
Yeah, dive in.
Dive in. Okay. So one of the things that I saw with writing this article, and that I found extremely interesting, was the difference between the expression levels of almost all the well-known genes of the innate antiviral immune system within the different patient groups. So just a little bit of background. I know probably everyone here knows the severity and the impact of this disease, and especially getting it as a child or adolescent, the amount of work, fear, and insecurity that comes with it. However, I would just kind of shortly touch upon the fact that the incidence of type 1 diabetes is increasing, and it cannot be explained only by genetics. The environment plays a huge role, and as you can kind of see on the graph on the right, especially in the Western world, the incidence is high to basically very high. And again, even though we know a lot about the genetics of the disease, the environment plays a bigger and bigger role of the pathogenesis, and especially such as viruses, enteroviruses, and also the intestinal microbiota, and possibly the permeability in the gut. Another thing that I actually found very important to kind of keep in mind, especially when going through the research and articles, is sometimes the lack of differentiation of the different types of diabetes. When we're talking about which type of diabetes, and basically that's because why that's important is, well, depending on the type of diabetes, the pathogenesis and the cause is different, therefore prevention and cure will be as well. And maybe even within type 1 diabetes, there might be multiple causes of the disease. Hence, it's really important to figure out what happens before the onset of the disease. So the pathogenesis of type 1 diabetes have 3 factors: the genetic the genetic predisposition and the environment, and of course also the mechanism of it, the priming of the autoreactive T cells that causes the destruction of beta cells. And I actually found this online from the Lancet, and I actually feel like this kind of gives a really great overview of what we already found out about type 1 diabetes causing the onset, but actually also how far we still have to go, because basically a lot of things can influence the disease at many different times during basically during your lifetime, and it's so important to actually kind of figure out what factor does what, when, and where. And we just talked about some of the things that especially Karsten have already contributed to the research of diabetes, and we also have some other things that we know can maybe reduce the risk of the autoimmunity. But it actually says it really well on the bottom part of this slide, is that the heterogeneity of type 1 diabetes might be explained by an individual's genetics and exposure to different environmental triggers at different life stages. And it is here where I think our paper actually comes in and gives us a really good idea where to start looking. So basically what you're seeing on this slide is the heat map from the paper, where if you actually are just kind of looking at it like an overview of it, it's actually interesting to see that, well, let me start over. Basically, we have 4 different groups of patients that are compared to a control group. So we have AB autoantibody positive, which is the first group. We have a small group of type 1 diabetes patient that has been newly diagnosed from the DVID study. And then we have a group of type 1 diabetes patient that have had diabetes for a longer period of time. And then we actually also had a group of type 2 diabetes patients, just to make sure that what we found was actually specific for type 1 diabetes. And one of the things that is kind of, it's a trend, it's not significant, but the trend is actually to seem to be that in the autoantibody positive patient, basically the pre-diabetic people, somehow or for some reason the innate anti-immune system is I don't want to say lazy, but it doesn't seem to be working as well as it's supposed to be. Whereas in the type 1 diabetes newly diagnosed, they're significantly upregulated, and actually slightly upregulated as well is in the patients with diabetes for a longer period of time. So basically, when you have something that is bluish, the gene is downregulated compared to the control group. If it's reddish, it's upregulated compared to the control group. And some of the genes that I kind of stumbled or kind of found extremely interesting was the receptors TLR4 and TLR7, and especially TLR7, because it's actually activated by enterovirus, which has already been associated with type 1 diabetes, and it's actually significantly downregulated in pre-diabetic patients and significantly upregulated in type 1 diabetes patients that is newly diagnosed. And both these receptors actually leads to activation of the innate antiviral immune system, and especially these genes called interferon stimulated genes, which are used in the cell to basically clear pathogens. And as you can see, it's not only just one pathway of these interferon stimulated genes, it's actually multiple of the bigger pathway of the ISGs that is significantly upregulated in type 1 diabetes, newly diagnosed type 1 diabetes. What made it even more interesting was that when we actually looked at SNPs, which is a single nucleotide polymorphism, basically just one change of a base in the DNA, we actually found a lot of SNPs, newly found SNPs that was associated to type 1 diabetes and placed within these same genes that we had seen dysregulated on the RNA level seen dysregulated at the RNA level in the islets of these different patient groups that we were looking at. So this is an overview of our data from the article, and it's a very technical figure. I'm not going to go into all the details. However, what is kind of important to notice that all these genes is basically involved in the antiviral response, or the programmed cell deaths, or the activation of the adaptive immune response. And if you're actually looking at it, the ones that are colored have some sort of changes to them compared to a control group. And when you're kind of looking at it, that's a lot of genes that they're not necessarily not working, but it also just seems like they might not be working optimally in the cells. For example, the beta cells, and the beta cells are already working hard at producing insulin to keep our blood glucose within range. So if you think about it, if these genes are already not completely working or not working the way that they were supposed to be working, it might not take that much of stress from the environment in some shape or form before it's actually too much for example, the beta cells. So what I kind of find interesting by our data is the fact that the innate antiviral immune system seems to be so involved in the onset, or in the lead up to the onset of diabetes. The immune system, the innate immune system, is activated within the first 0 to 24 hours of anything kind of entering your system and causing some sort of inflammation. And the thing that is really important with a disease like type 1 diabetes is that to figure out when and how early all these changes actually happen. If we want to somehow prevent it or cure it, we need to figure out how early all these changes that causes the disease, that causes basically normal working islets to suddenly be pushed into a pre-diabetic phase, where suddenly in the periphery, basically cells, autoaggressive cells or T cells, starting to kind of be in the area of the islets and infiltrate them, and basically in the end we end up with destruction of beta cells and symptomatic type 1 diabetes. So the earlier we can kind of figure out when this innate immune system actually starts to have an impact, the better, because that also will help us figure out, well, when would preventive measures have the best impact? And it's definitely one of those things where preventing something from breaking, instead maybe of trying to fix something that is already broken, might be a way of doing it. Sometimes we also need to be able to fix something that is broken by understanding why it's breaking in the first place. So we really need to figure out where the biggest impact of the genetics, of the environmental factors are coming, and basically before that can happen, we need to have more knowledge about the interplay between the different organs in the pancreas. We need to understand the genetics and the environmental factors and how they're causing the type 1 diabetes, and the different pathways involved and their interaction with each other. And again, the big question is, why are the beta cells so vulnerable? And again, when can we actually have the biggest impact with preventive measures, especially like fenofibrate that we were talking about? Could we maybe see if we could catch people earlier, maybe it would have an even bigger impact. So could we find some other things that could maybe help out in the womb, if we could somehow predict who may be predisposed to developing type 1 diabetes, or even when our islets are still working the way that they're supposed to be working, somehow help these individuals that are predisposed to keep on having a pancreas that is working the way that it's supposed to be working. And I do actually believe that the paper that we came out with is a very good starting point of figuring some of all of these things out. And with that, I just want to say thank you to all the people that was involved in making this happen.
That's fantastic. I wanted to just see if we could talk a little bit about the genes that you did find in your March 2021 Diabiologia paper. You found that the most aberrant genes were in a group of interferon stimulated genes. OAS1 increased by 111%, MX1 increased by 142%, and ISG15 increased by 197%. If we talk a little bit about what is ISG15 associated with in a normal state? What is it doing?
So basically all of these different pathways, the 2,5A pathway, which consists of the OAS and RNA cell, the MX proteins, the ISG15, they all have different roles in trying to avoid or trying to stop the virus when it's kind of entering the cell.
And are they sequential in their actions or no, or is it all at once?
It's basically depending on where in the cycle of or where in the life cycle of the virus. So I'm not a biologist, so bear with me. But basically, as far as I remember, the MX proteins are part of the whole packing up the virus or the entry of the virus, trying to stop the virus there, where the ISG15 is in trying to basically, again, avoid the whole RNA of or the protein or basically avoid the virus to transcribe the proteins that they need. And kind of target them for degradation. And where the OAS RNA cell pathway is kind of a guard dog in the cytoplasm of the cell, where they kind of, especially OAS, kind of senses if any double-stranded RNA appears, because it's an unnatural thing to be present in the cell.
Yeah.
So it can activate RNA cell that then can go in and actually cleave the virus single-stranded RNA from the virus, or even the host's own single-stranded RNA.
Can you speculate why someone who has T1D for a long time does not have the upregulation of these genes?
Well, to be honest, it kind of makes sense, because at some point you kind of see the system reach some sort of a normal level, where the high upregulation that we might see in the type 1 diabetes newly diagnosed, I definitely do believe that the whole disease might be activated or somehow aggravated from a virus. But why we see them still being upregulated might also just be because these genes are also part of the programmed cell death. So we might also see upregulated because the beta cells are basically breaking. The other thing could also, the other thing just disappeared from my brain that I was about to say, but the other thing could also basically be like there is some sort of virus present, but I think the prolonged increase of them might just be because we're seeing the beta cells breaking.
Yeah, it also kind of is curious, like in antibody positive individuals, you don't see that same signature. It almost suggests like there's a real discrete window, which is really, companies like Prevention Bio are kind of going after that window to, stop the actual infection of the beta cells by the virus. So I think that this is really great that you guys have, identified this sort of window of activity. And I didn't know if Karsten, do you want to weigh in here on the topic?
Yeah, I should mention that for years ago, we have published that this 2,5 synthase system is 6 times more active in beta cells than in alpha cells. and this could be an explanation. I'm not saying it's the explanation, but it could be an explanation why the beta cells are the one that are destroyed at the very end by T cells and not the alpha cells.
Mm-hmm.
It's mentioned in this paper as well, but it's published for.
So the
some
2,5 pathway is more active in beta cells than in alphas?
Yes, that's right. 6 times we have found.
Oh, 6 times, yeah. And so that is interesting. How might we start to think about that, you know, the environmental insults you mentioned, you know, cow's milk, gluten. I mean, type 1 diabetes is on the rise, right? And then so we think, oh, maybe there are some environmental changes, you know, post-industrialism, it seems to be on the rise. Type 1 diabetes seems to be on the rise, and maybe there's impact in our, I guess, cow's milk, gluten. How might that be different and interacting with beta cells than it did historically? Because, I mean, it is interesting. There's a large incidence of type 1 diabetes in the Scandinavian countries, and those countries are, the diet is very cow's milk based, isn't it?
We definitely do like our milk and our milk products.
Yeah. But how could that have changed? I mean, that's you know, that the evolution of eating, and you know, there's just a science article about how, you know, how the humans and cows, the milk sort of evolved together. But it's like, why did that? Why wasn't there more incidence of type 1 diabetes seen earlier in this kind of historical diet?
Well, one of my ideas is definitely that probably the way that we're processing things can have an impact on it. But it's also one of those things where maybe it's not just one thing that it's the cause. Maybe it's multiple things that's the cause of it.
Yeah, like the cow's milk and the gluten, sort of environmental things set it up, and then when a virus comes in, this window, you know, something.
Yeah, maybe with a touch of the hygiene hypothesis of it all. I mean, it's one of those things, again, to back to the heat map, where it's kind of interesting that it seems like there's a tendency of a trend of the immune system kind of being slow or lazy. So I mean, maybe it's also a genetic evolution that we haven't found yet. I mean, that's also one of the things that was so interesting in the paper that we found so many new SNPs, and it could also maybe be some epigenetics of it all that haven't been taken into consideration yet. It's definitely one of the things where I'm thinking we know a lot about the disease, but I also think there's a lot of things we don't know yet. And I think more biomarkers, and especially within the innate immune system, could be of interest. The other thing is looking at the interaction between, for example, the gut and the pancreas could be of interest, of trying to explain what we are seeing.
Yeah, I mean, there definitely is an interaction there. I wondered if, Karsten, if you could comment on the sphingomyelin. If we talk about sphingomyelin and, you know, how it may impact beta cells, could there be a way that sphingomyelin protects from viral, you know, from viral assault on beta cells in some manner? I mean, is there a way to track whether or not in the presence of sphingomyelin that these genes, these dysregulated islet genes, you know, don't get so dysregulated? I don't know, just sort of throwing it out there.
Yeah, it could influence the stress of beta cells, and we are thinking on that at the moment. I should mention the cleanness of the surroundings. You know, in Karelian, which is divided between Finland and Russia, in the Russian side, they are not that clean. In Finland, they're extremely clean, and the incidence is up to 5 times of diabetes higher in Finland than in Russia. And also in Denmark and US, of course, there's clean and maybe too clean. But that's not easy to explain, especially not for the female part of the population. They want, yeah, as you know, things should be clean, clean, clean. And that's maybe not physiological, and we are from the Stone Age where everything was much more dirty. So I think that's a really main factor. And yeah, the sphingolitis, I hope we come up with some more new things, as I told you before, about how it's acting, and that's an exciting area at the moment, I should say.
I wondered if we could open it up, excuse me, to some questions from the audience.
Yes, that's fine.
Feel free to drop something in the chat if you don't want to speak aloud. I would also, while we're waiting, how about, you know, when you found the 27 novel SNPs, you know, how might those SNPs, that larger group, be impacting the phenotype?
Yeah, that's gonna be my work for the next couple of years. Because we do actually, that is the next big question, is like, how are they actually impacting the phenotype of the genes? Do they actually have a direct impact on the genes, like the actual protein, or is it something, could it actually be something to do with changing the epigenetics of the gene within the promoter region, which is gonna be, it's gonna be so interesting to look into, and yeah, there's definitely a lot of work for me for the next couple of years to try and figure that one out.
Yeah,
but
we just spoke to Steve Rich at UVA recently, and he said that a lot of the genes that they've seen that to be dysregulated, right, are not in coding regions.
Yeah.
So
and that's also, I mean, as I was saying, I've been working with at least the 2,5A system for a long time, and I mean, SNPs are extremely interesting to look at, but sometimes it's not always that easy to see how they actually have an impact. So even though we might have found these huge amount of genes, of SNPs, it might not actually be all of them that have a direct impact on the disease. However, I'm really interested in looking in the ones that we found in the OAS family, just upstream of the transcription start site, especially in combination with some other SNPs that other groups have shown within the OAS genes, because I know basically, because because all of these pathways, the interferon stimulated genes, are so important for the cell and upholding the cell's viability. They're so well regulated, and especially in the OAS 1, 2, and 3, there's actually a huge difference. Even though they're relatively similar, there's actually a huge difference in the way that they're reacting, or what they're contributing to within the cells. So it's going to be interesting to look into that, but it is some of the work that needs to be done.
Can I also ask, you know, for OAS 1, MX1, and ISG15, the three that were really most aberrant, are those differences seen in other autoimmune diseases? Some of the cousin diseases, like, you know, MS, RA, lupus, etc.
Yeah, I don't necessarily have seen ISG15, but MX, the MX proteins and OAS have definitely been involved in other. Actually, in my old lab, despite the fact that we were in my old lab where we work with virus-induced cancers, we looked at the 2,5A system in colitis, and they were also involved in basically the permeability of that disease.
Mhm.
So it's definitely, it makes sense, but it's also a pathway that's extremely hard to work with. So and even with the MX proteins and the ISG15, there's actually not that much research out there because they are extremely hard to work with. So you kind of have to be a little bit crazy to to keep working on them.
You mean that just the protocols are finicky?
Yeah, and they're just so difficult to actually get expressed on protein level. For example, the OAS genes, despite the fact that they might actually be that we actually saw that they were extremely dysregulated, for some reason we didn't see RNase-L, which is basically dependent on the OAS genes to get activated. That wasn't significantly regulated, dysregulated. So that's one of the things that is difficult to work with. The other thing is just to get it to express on protein level, because basically the 2,5-A pathway, it within hours it can kind of go to the max, and then within half an hour be down. So it's kind of like, it basically goes like a wave. So it can be difficult to catch it at the right time.
Yeah, to really detect it.
Yeah.
I'm curious as to whether or not anyone's done work on these three genes in context of COVID.
Oh, they have.
Yeah, I'm sure. Yeah, I mean, so
Yeah,
maybe that'll give a repository of data that can be tapped into and viewed in context of type 1 diabetes.
Yeah, it's definitely something that I'm keeping my eye on.
I'm sure. This has been so fascinating. I really enjoyed this, and thank you so much to both of you, Drs. Pedersen and Buschard. We're really interested to see what comes next, and also to keep an eye on the patient whose treatment with fenofibrate by Dr. Buschard has kept her in remission for 3 years. So, this is very not unusual, but it's definitely something to watch, and I feel like there needs to be more of a spotlight on it. I would also encourage and invite everyone in your realm over in Copenhagen at Rigshospitalet, and to join us in keeping your eye on what we're doing here at the D Challenge. We have a consensome. We're offering it to people to mine, and we're hoping that that we'll be able to kind of pull some interesting genes out of this consensome, which has been developed and created by the Signaling Pathways Project, led by Neil McKenna at Baylor Medicine. So sort of exciting times in the world of genetics, and we appreciate all you're doing in that field. So thank you again for talking to us.
Thank you so much for having us.
Have a great rest of your day.